You can look through that on your own. It's a summary statement, and the only two things in this thing that don't change are those things. Everything else can change. All right, so a few common patterns in adaptation, and then we'll start to talk specifically about aerobic adaptation. If you remove physical activity, people undergo atrophy or detraining. Those of you who are ill, if you've started to try to train again, it's always shocking that if you haven't exercised for a week or two, it's like, what just happened? And you will lose things. You will lose sarcomers. You will lose mitochondria. You will lose enzymes. If one is doing heavy resistance training, the tendency, because the limiting stress when you do this is maximal force, you tend to get a focus on the gene expression on sarcomeric proteins. You add sarcomeres in parallel. This increases the size of the myofibrils and therefore increase your maximum force. It's very interesting. There's almost no change in metabolic enzymes. Remember, metabolism is all about rate of replacement through time. And so if you're just doing a kind of 1 rep max, it's almost not metabolic. I don't know how to say that. You're not going to run out of ATP in 3 seconds. And so it's interesting that you rarely see large-scale changes in metabolism. Although we did see in people doing kind of standard style weightlifting, the fast twitch fiber, because it's being recruited, actually becomes a little bit more aerobic, and that's mostly because we tend to do multiple sets. So you actually are asking it to pay off its oxygen deficit at the end of each set, and so they actually become a little more aerobic. Short-term high-intensity work, what is sometimes called high-power events lasting, these are going to be based on cytosolic enzymes of glycolysis and the phosphagens, and that's what you'll see adapt. And finally, this is the one way back in the 80s, or 70s, the term aerobics was formed. First recognized, this is the first kind of training fitness that was recognized. That, hey, they're adding mitochondria, they're adding oxidative capacity, the measurable VO2 max, the time to exhaustion, all these things are increasing with endurance training. Does that all sound okay? As a general? Okay. This is sad but true. So here's weeks of training. You ought to change, if you have a chance, put a line through this and write months of training. This is just too optimistic a textbook figure. And they're showing mitochondrial content. If you want to double your mitochondrial content, you're talking 6 months, to be quite honest, of hard training. So that'd be more. But what this figure is supposed to show you is that the adaptations due to training depend on the recurrence of the training stimulus. So if you stop, you can never raise your hand and say, I have achieved fitness, I am done, because it leaks away. And one of the saddest things is the slope of the leakage. See this as it drops? You will lose, let's go from here. So that is about there. It looks like about something like 4 to 5 months of gain is lost in something like 2 weeks of detraining. It's usually about 4 to 1. In 1 week, you lose what it took you 4 weeks to gain. And that number varies depending on which attribute you're talking about. But almost always the rate of loss is faster than the rate of gain. One way to think about this is these adaptations are expensive, right? To maintain a muscle mass, to maintain protein, to install mitochondria. This requires energy. And so if the need to have it stops, you remove it. It's sort of like the world's most effective housekeeper. If something hasn't been used in the house in the last week, she tosses it out. No, you can't have it anymore. And so it's unfortunate but true, and one of the few drawbacks to exercise intervention as a health, exercise intervention is the most powerful health benefit you can have. The bad news is it's lifetime therapy. You don't get stopped. Okay? So now the rest of the time, good time for questions if you'd like. I was going to talk for the rest of today's lecture about the adaptation to endurance work. And you'll see you know a lot of this already. Any questions? Good. Okay. So this is, what do we call this where the amount of force you get as a percentage of maximum force generation is a function of how much of the motor pool you recruit, with each one of these little things being a single motor unit. What do we call that relation? The size principle. It's really important. And notice, this used to shock me, that when you're walking, you've probably recruited 10 to 15% of your total muscle mass. Or at least a better way to say that is, the amount of force you're using as a percentage of maximum is about 10 or 15, and you may have recruited 40% of the muscle mass, all of the slow twitches in your muscle. Notice that even when you run, you tend to barely get through all your slow oxidatives and your intermediate fatigue-resistant fibers, and in a sprint, you're just barely starting to touch your fast glycolytic fibers. It's worth noting that, it's just interesting. Now remember, the metabolic characteristics and the fatigability of these fibers can be altered by recruiting them and asking them to practice whatever the task is. Why did I put that up? So we're going to focus down here. Oh, I know. That means that if you go out and for your workout, all you do is trot. So I'm going to put the line for trotting right about here, 20% of maximum voluntary contraction. What fraction of your muscle doesn't even turn on? Something like 45% of your muscle is not even being exposed to the activity. This is one of the reasons why a good training program often complements weightlifting with aerobics, because weightlifting will recruit every muscle in the, or every cell in the muscle if you're going to fatigue. But these aerobic adaptations are the one that have the greatest health benefits. So you kind of want it all. You can look through that on your own. It's just reminding you of the structural, the different types of fiber. So, how does metabolic adaptation occur? Remember, many of you asked, how do you change the Vmax? And the most common way in a biological system that you change the Vmax, you add more enzyme to the system. You simply put in more enzyme. If here you're saturating at 1500 whatever enzyme slots, and you up this to 2000, well, you just increase the capacity of the reaction by 25%. And so by far the most common way that we get adaptation in muscles is, let's add more pyruvate dehydrogenase. Let's add more carnitine and carnitine acyltransferase. Let's add more enzymes of the TCA cycle. And automatically, you have a system with a higher capacity for ATP replenishment. Does that make sense? So simply express those genes more. And there's just huge amounts of information going all the way back to when I was a puppy. We first discovered this. So here's carnitine palmitoyltransferase, and here's succinyl dehydrogenase. Well, I can identify both of these as being mitochondria. I understand that. Notice that when you do aerobic training, you get large-scale increases. Here's almost a doubling. Here is more than a doubling when you do purposeful anaerobic training. Notice that when you do malate dehydrogenase, let's pick something that's a little bit more glycolytic. Let's pick phosphofructokinase. That's clearly in the glycolytic pathway, and phosphorylase. Notice that phosphofructokinase really goes up primarily not with aerobic training, but with anaerobic repetitive sprint training. So your enzyme, your metabolic enzyme adaptation tracks whatever the contractile task is. What's the limiting feature here? That's what you're going to enhance. Does that make sense? There's so many examples. I could just do this forever. So hexokinase, in terms of importing, this is finally a decent figure. Look at this. We've got the capillary, we've got the exiting, we have the interstitial space represented, and we have the GLUT4 receptor. Finally. You could add more GLUT4 receptors. Remember that they're upregulated by the presence of calcium. You could actually express more GLUT4 receptors in the recovery period and have bigger vesicles with more capacity for transport. And it is certainly true that you add more hexokinase. How long is this? Sorry, I don't have the period of time. I think it's a short college student study where, so it's about 8 weeks. And so within 2 months, these people have basically added 50% to their hexokinase ability to import glucose from the bloodstream. It's not bad. The same is true of fat oxidation. This is all aerobic training. Notice that, what is this? The plasma free fatty acid concentration after training. These are going to be, this looks like duration of fixed work. So this is duration. Notice that after training you get higher levels, so you're actually recruiting more free fatty acid into the bloodstream. That suggests that hormone-sensitive lipase activation in the adipose cells themselves has been enhanced. Notice free fatty uptake by the muscle itself. That suggests that fatty acid binding protein and the FAT protein are both enhanced at the surface of the muscle cell. And then here's, well, this is fun, recruitment from the adipose tissue, uptake by the skeletal muscle, and then oxidation within the skeletal muscles mitochondria. Notice, by the way, you have some of the Randall effect. Where does this change the biggest? See how it's biggest over here, and so you're seeing, because they must have picked a fixed workload around 60% of VO2 max, the longer they go beyond 90 minutes, the more you see the shift, really demonstrating just how much more mitochondrial mass is present and capable. Does that make sense? By the way, just as an aside, since we're talking about glycogen sparing, if this is how much energy was being derived from fat before training, what does that mean about the time to glycogen depletion before training? It's going to be much sooner because you're not using, what if you, this to me looks like a doubling, 50 to more than 100. If you've doubled the amount of kilocalories coming from fat, what does this mean about your glycogen stores? You're sparing them. See what I mean? They're still there, and that leads to the next figure, which is that the training adaptation, here's the energy from carbohydrates during those same prolonged workout. Notice how the energy from carbohydrates is actually falling off, especially towards the end. This is the energy from fat, and this is the training effects of the fat. Oxidation has gone up, sparing the glycogen. And what that tends to mean is the time to glycogen depletion goes way up. So here's muscle glycogen. Let's see if we can figure this out. After training, decrease. So before exercise to after exercise, they went from, this is the same workout. They used 200 of whatever these units are, millimoles per kilogram. They used 200 by the time they quit. Notice this is after they've trained. First off, they've added more glycogen. And they've hit it, what's 200 off of that would be 228. They've actually used more. Do I have that right? No, here they use 200. Here they're only using 100. So they've actually reduced their use of glycogen, even though they have more to start with. Does everyone start to see how if you've doubled your total glycogen content, but you're using it only half as fast, that suddenly your time to exhaustion may be fourfold? If before you exhausted at 1 hour, you can now run a 4-hour marathon. Is that making sense? It's very cool, very exciting. Yeah.
Does that also mean you can go hungry for like longer?
Yes, absolutely. The exercise state institutes a lot of protections against other stressors, and so I think that was Tremay's voice. My eyes are shut. Suggest that, for instance, is Ramadan more tolerable if you have, yes, it is, yes. Is fasting, can you go longer before you actually dump your blood? Yes. All of those things. Let's see. So, we talked about this. Notice again, down here, we're talking about the intensity of work. And so our crossover, it's an idealized graph. The crossover is the harder you work, the less fat you use, and the more carbohydrate you use. What would the training effects that we've just looked at lead to, as far as where this crossover is? It's going to go out further, right? So if the crossover point for this person is down around 50% VO2 max, the crossover point will actually move to a higher intensity before you become, if you want to say it, carbohydrate dependent as a fuel source. This is more of the same. This is Golnick, Saltin and Golnick's famous paper showing succinyl dehydrogenase. So succinyl dehydrogenase is a mitochondrial enzyme. This is enzyme activity. So these aren't percent of type 1, percent of type. This is showing you how aerobic is each of these cell populations. And in the untrained person, oh sorry, here we got deconditioned, sedentary. And then we have conditioning. Notice that the average folk here, if they undergo training, they add succinyl dehydrogenase activity, especially the highly conditioned athletes. They add activity in their type 1 or aerobic cells. Notice the type 2s. This is interesting. I want you to look closely because this is where we'll finish today. One more intellectual challenge for you. Notice that those who are doing just sort of standard PE class conditioning, in the type 2As, they increase their succinyl dehydrogenase activity. But if you go to their type 2Xs, they really don't. Whereas, if you look at the elite athletes who are training for competition, they actually get a succinyl dehydrogenase adaptation even in their 2, by the way, their 2Xs probably become 2As at that point. But the original cells that were 2Xs add a huge amount of succinyl. Anyone want to tell me what the difference is here between the regular trainers, the routine trainers versus here?
Are the endurance athletes able to recruit?
They're actually recruiting these fibers because they're running, as we say in Northern California, hella fast. These folks are trotting, and so they never recruited those fibers. Here is another study. Oh, I had some Spanish. I thought I had Spanish. Okay. Here's the same data graphed, and it's showing you that in the aerobic muscle, so these are probably rats in this particular study, if you take the aerobic fraction of the gastrocnemius, that 30 minutes of exercise, regardless of the intensity, the entire muscle is recruited, and therefore you get most of the change in citrate synthase comes simply from turning on the muscle. You get a little bit bigger effect if you up the workload, but mostly if it's recruited, it will adapt. Now if you look at the fast twitch, oh yeah, 2B, so this has got to be rats. If you take the white element, the white half of the gastrocnemius in rats, they're more differentiated. Notice that you get almost no change when you're doing light activity because the cell never turned on. You pump the activity up to 65%, you start to get some adaptation. But if you really want to get adaptation in a fast twitch fiber, you better work it hella hard for hella long. And then you actually begin to get adaptation in an aerobic enzyme. So again, only fibers that are recruited will adapt. You got to invite them to the party before they'll dance. Is that okay? That's a hard figure. But think about it. That's all it's saying. Now the last three figures are really straightforward. I didn't think I'd get this far, but it's wonderful. This is a textbook figure just pointing out, if you do this aerobic training, the time to fatigue, so this is, let's see, if you're fatiguing in 8 minutes, this is probably 95% of VO2 max. So they put the person at very high oxidative level of work. And we see that slowly they fall off, but they produce more work after training, and they fall off less in their work output. So the fibers that you have recently recruited and they've practiced using oxygen, they become fatigue resistant. In other words, your FFs, your fast fatiguables, become more FR, fatigue resistant, fast fatigue resistant. Your lactate threshold tends to shift. Workloads that previously caused you to take off, if this was 180 watts and that's 200 watts, and you get to 200 watts and you start to take off, notice that now you can tolerate 200 watts and perhaps even 300 watts before you start to move into the large-scale recruitment of fibers that produce lactate. So these two figures are not unrelated. That and that are related because they're both talking about the larger motor units that are usually a little bit more glycolytic. They've practiced, and now they're less fatigable and more oxidative. We'll talk in a week, I think, about the cardiovascular delivery of oxygen. So that'll be things like increased cardiac output and increased blood flow. So that's presentation. The bloodstream is like a waiter with a tray of hors d'oeuvres going, would you like some oxygen? Would you like some oxygen? The point is, you can't clear the hors d'oeuvres off the tray unless you have a hungry audience. And so that is, the muscle cells themselves have to have an enhanced ability to pull oxygen out of the bloodstream. And that has a huge amount to do with myoglobin content, mitochondrial content, and capillarity on the surface of the cells. And all those things are adaptations. So this is actually showing you the extractive ability. This Q dot VO2 max is showing you the muscle tissue itself, independent of the cardiovascular system, its ability to extract oxygen. It's gone way up. It's become a vacuum cleaner for oxygen. I know what to do with that. Is that okay? Oh, I should have, here, before I ruin that. So make me a proposal. We've seen at the beginning of class that the mitochondria are a bit sluggish, being deep inside the cell and insulated by another two membranes. They're slow to rise with the onset of work. They thus create the oxygen deficit by their slaggardness. That's a word. Tell me what's going to happen after these adapt. What's going to happen to the oxygen deficit in an adapted person? It becomes much smaller, right? They can accelerate their mitochondria faster. Many of you notice that second wind phenomenon when you first start running and it's all ugly, and then you get maybe 5 minutes in and you finally kind of go, oh, I might live, right? And you'll notice that the better shape you get in, the more rapid that experience of I'm going to live occurs because you're establishing the new steady state for oxygen transport much more quickly. So the oxygen deficit would be the difference between these lines and a straight line coming across here. Hey, by the way, remember how we said efficiency doesn't change? If efficiency changed, the total oxygen cost of the workload would change. So it's not the efficiency that's changing, but the ability of the mitochondria to accelerate themselves and cover the workload oxidatively becomes better and better and better. All make sense? Great. So we're all caught up. I have an office hour. I will try to post you some review materials for later this